Alright, so today we're going to talk about protein synthesis. Synthesis means the creation of. So protein synthesis is going to be creating proteins. Our ultimate goal is to figure out how a protein is built. It's important to figure out why a protein is built because what we're going to see is that a protein determines our traits. So it determines our physical traits. We're going to talk more about that down the road when we talk about genetics. But for now, we need to know that how a protein is made is really significant because proteins are important. Alright, so right here we got one nice piece of double-stranded DNA. I'm putting a pink little box around one gene. A gene is a sequence of DNA that has a message, and that message is how to build protein. Well, here you see there are multiple genes on DNA. In fact, there are millions of genes on DNA because our body needs millions of different proteins. Each gene controls a different trait. Each gene contains a different trait, like I said, so you can see there are multiple genes here. You can see that they're on both sides of the DNA, but they don't overlap each other on the same side, and they don't overlap to both sides. So we have 6 different genes here: eye color, unibrow, toe hair, hair color, freckles, and ear size. Those are 6 completely different genes. 3 of them are on one side, 3 of them are on the other side. They overlap, like hair color and unibrow overlap if they're on opposite sides, but they don't overlap on the same side. So eye color and unibrow don't overlap. You can see that each one stays on one side or the other of the DNA. So a gene's always found on one side or the other. Today we're going to follow the unibrow gene and see how we make that protein that says, hey, you've got one big giant unibrow. So the whole reason why I just showed you about DNA when we're talking about protein synthesis is that DNA is broken up into these things called genes. Those genes get copied into something called a messenger RNA or mRNA, which eventually tells us how to build a protein, which eventually gives us our trait. The DNA or the gene has the instructions for how to build that protein that's going to give us the trait. The problem is DNA is huge. It's a double-stranded molecule. It's super long. It's made up of many genes. And it's always found in the nucleus. So there's no easy way to get DNA out to where a protein is built, which is the ribosome. DNA can't easily escape the nucleus. So we have to find an alternative way to get the message out of the nucleus into the ribosome of how we need to build a protein. That alternative way is mRNA. mRNA stands for messenger RNA. It's just going to be a little copy of the gene's message in RNA form. It's small enough that it can fit right out of the nucleus through something called the nuclear pore, and it can get to the ribosome and carry the message of how to build that protein that's going to give us the trait of a unibrow. There's 2 huge major steps to protein synthesis: transcription and translation. Transcription is going to be pretty simple. It's going to take the DNA and turn it into mRNA. And translation is going to take that mRNA and it's going to actually get the code out and build a protein based off of that. How it's going to build the protein is using amino acids. So if you remember, amino acids are the monomer or individual part that makes up a protein. So when you get a bunch of amino acids together, you've got a protein. Okay, so here you have a cell. We're going to talk about transcription. This is the gray part's the cell membrane on the outside there, and then the purple is the nucleus. So inside the nucleus, we have DNA. We know DNA is in a double helix form. Well, we are only using a little bit of the DNA today. We're only using the unibrow gene. Whenever we're working with the double helix, the first thing we have to do with the double helix is unwind it, unzip it, so that we can get into the nitrogen bases to copy them. Helicase is the molecule that unwinds and unzips our DNA. So remember, helicase is an enzyme. Sounds like helicopter. Woo, woo, woo. Right? Next, we got to build the mRNA. So we have a molecule called RNA polymerase. RNA polymerase goes out and it finds the free-floating nucleotides that pair up with our DNA nucleotides to build the mRNA. If you remember from what we talked about in class, an mRNA, or an RNA in general, they still have adenine, they still have guanine, they still have cytosine, but they have a 4th different base from DNA. Whereas DNA had thymine, which was T, RNA has uracil, which is U. And uracil pairs with adenine instead of thymine pairing with adenine. So when we build the complementary mRNA, it uses U. So RNA polymerase puts together the mRNA based off of the DNA of that gene only. So the mRNA is way shorter than our DNA because it's only the length of the gene. So it's only the length of the purple part there. Our mRNA now has the message of how to build the unibrow protein. We just copied it into it. All it's got to do now is take that message to the ribosome so the ribosome can actually build the protein. So the mRNA snakes itself out of the nucleus through a hole called the nuclear pore, and it heads off to the ribosome out in the cytoplasm, and that's going to begin the second part of our protein synthesis process called translation. So again, transcription, you unwind the DNA using helicase, you unzip it using helicase, you copy the one gene you want using polymerase and free-floating nucleotides into mRNA, that's transcribing, and then that mRNA leaves the nucleus for the ribosomes to start translation. Alright, so here we're at the ribosome, the black is the ribosome, in comes mRNA, look at him swimming on in, he's arriving at the ribosome just now. Our ribosome starts reading, and what it's looking for is something called the start codon. So it would start off and it would read A, C, C, nope, G, nope, A, ooh, that sounds good, U, yep, G, A, U, G, that's the start codon. And so it calls down the first molecule. Those little molecules that kind of look like cactuses that are flying in right now are called tRNA. The tRNA carries the amino acid. So the tRNA stands for transfer RNA. What is transferring is the part that is used to build the protein called an amino acid. The tRNA arrives and it brings in that amino acid, and then our ribosome reads the next 3 bases. Our ribosome likes to read by 3s. Every 3 bases that it reads is called a codon. So when it reads the next 3 bases, it calls in another tRNA. That other tRNA comes in and it brings the corresponding amino acid with it. When that amino acid arrives, it bonds with the amino acids that were already there, making a longer chain of amino acids, and the first tRNA leaves to go recharge itself. The ribosome can only hold 2 tRNA at once, so this process is kind of slow because one comes in, one leaves. One comes in, one leaves. Each tRNA carries in a unique amino acid. How we determine what tRNA needs to come is based off the anticodon on the tRNA. The anticodon on the tRNA is this part right there. Right? That one's GGG. The anticodon pairs up with the codon. So our codon is on the mRNA, CCC. They're opposite each other. Just like mRNA, messenger RNA, tRNA, transfer RNA, is RNA, which means it's going to have uracil, not thymine, as one of the bases. Alright, so that tRNA would come down and it would bring the amino acid with it, and we keep prolonging this chain and lengthening the chain. That's good because what we're trying to build is a big long chain of amino acids which will get folded into a protein. Alright, so the ribosome shifts down, reads the next codon CCC, that sends out a signal for the tRNA that has GGG as its anticodon. That one's bringing proline amino acid. Proline amino acid bonds with histidine amino acid, that's a peptide bond, and the first tRNA leaves. And then we read GAG, CAC comes, the CAC tRNA comes down, it brings glutamate, they bond, and the first tRNA leaves. And our ribosome shifts down again, it reads UUA, sends out a signal for the tRNA with the anticodon AAU, the opposite that pairs with it. That one brings leucine, they pair up, they form a peptide bond, and the first tRNA leaves. Again, our ribosome shifts down, sends out a signal, calls the next tRNA, it brings the next amino acid, and this process keeps going, and we're going to make a really long amino acid or chain of amino acids. Now, what's different here is we've reached what's called the stop codon. The stop codon sends out a signal for us to stop building the protein. What's different about the stop codon, as you can see, is that its tRNA doesn't have an amino acid. That means when it's time to pass the chain on, nothing can grab it. So our chain is released from the ribosome disassembles, and we have this big long chain of amino acids that's almost a protein. It's called a polypeptide chain. The reason why it's called a polypeptide chain is because the bonds between the amino acids are called peptide bonds. So poly means many, and then peptide is the bonds between the amino acids. So we have many bonds between amino acids here. So it's a polypeptide. That polypeptide is almost a protein. It's so close it can taste it. The only thing that needs to happen with it is that it needs to be folded up. So here we go into our last step of protein synthesis. After we've made this polypeptide, this big long chain of polypeptide is going to swim on down, and it is going to go into what's called the rough endoplasmic reticulum. So you can see it's entering the rough endoplasmic reticulum. Here it's going to be folded. So when it's done at the rough ER and the Golgi apparatus, our protein comes out nice and folded, and now it can work. It's a protein. So that's actually going to give you your trait. It's actually going to give you one giant big unibrow, which is really important that we made the right protein. So it's going to give us our trait, which is the unibrow trait. And that, in a nutshell, is protein synthesis. We've got transcription and translation. Transcription, remember, we get the message from the DNA, turn it into messenger RNA. Then that messenger RNA carries the message of how to build the protein from the DNA, clear out of the nucleus to the ribosome. At the ribosome, it gets built. The protein gets built using amino acids starting at the start codon, AUG, and ending at the stop codon, which calls for a tRNA that brings no amino acid, causing a stop to our amino acid chain, which then gets folded into a protein. The end. See you next time.